Okay, welcome to, I believe this is the fourth part of DNA and RNA replication, transcription, and translation. We talked about what DNA and RNA is. We talked about the roles of DNA and RNA. We talked about the replication of DNA in the nucleus before mitosis or mitotic divisions. We also talked about what happens and the process whereby DNA is transcribed into RNA material so that that material can be processed to leave the nucleus. And the reason why the RNA, the mRNA, needs to be processed is because we want to make it ready to make polypeptides, which will then be making proteins later on. These proteins then will catalyze the reactions necessary for making everything that we need in the cell. So remember that not everything that we need in the cell is present all the time. In order for some things to be present, we have to transcribe and translate them into proteins, and then from then we can get the things that we need. So we talked about transcription. We talked about the copying of template DNA by RNA polymerase to make a strand of pre-mRNA. This pre-mRNA is then processed. The introns are taken out. The exons are fused together. We add a 5' cap and a 3' poly A tail to make it possible for this molecule to exit the nucleus and go into the cytosol without being broken down. It also acts to message that messenger RNA to the right place. That's what the 5' cap and 3' poly A tail. We also talked a little bit about alternative splicing and how you can take pieces of exon and just rearrange them, and we can end up making different proteins and products from them just by rearranging the exons, and that's called alternative splicing. All this is done by a spliceosome. But the focus of this part is translation. So transcribing means to rewrite. To translate means to change language. All right. So what we're doing here is we're changing nucleotide language to amino acid polypeptide language, whereas DNA to RNA is changing one nucleotide form of language and changing it over to another nucleotide form of language. In this process, we're taking nucleotide language in the form of RNA and we're translating it to polypeptide language. So this process is where we're actually going to take the language of the RNA and we're going to change it into polypeptides. So it's really exciting, super exciting. Translation occurs in the cytosol of cells. It involves the messenger RNA, which is now a mature messenger RNA. We have a new RNA called tRNAs, which are called transfer RNAs, and they're going to be involved with reading the mRNA language and matching the codons with something called an anticodon. And each anticodon actually has its own specific amino acid that it codes for. So the language of the RNA is going to be translated into a language of amino acids by codons, or triplet codes, which we talked about earlier. And one of the most important things going on here is the rRNA, or the ribosomal RNA. Ribosomes have two big subunits, or two subunits, a big and a small subunit, and what happens is that the small and large subunit actually attach together to form one big subunit with the mRNA in between, and that's how translation begins. There are 3 sites on the rRNA. The ribosomal RNA has 3 sites. The first one is the active site, A site. The second one is the P site, and the third one is the E site. Remember here, something a little bit different here is we're going to be reading the mRNA strand in the 5' to 3' direction. So where we make DNA and RNA, reading in the 3 to 5 direction, and we make in the 5 to 3, in translation, we're actually going to be reading the mRNA strand starting on the 5' end and moving in the 3' direction. So that's one change from before. We have things called codons, which are a triplet code, like AUG is a codon, CCC, GGG, AAA, ACG. All those are codons, and we're matching them with anticodons, which complement the codons and actually are associated with a specific amino acid. So translation is an awesome process. Can't wait to tell you about it. Remember, this right now, sorry, it's the morning, early, and my mind isn't working as well. You're going to need to watch other videos. You're going to have to ask questions in class. I'm going to be writing this whole process up on the board. You'll have a chance to ask questions. Draw it yourself. Look up in your textbook. There's plenty of information in your textbook about it. This is designed for you to get a very thorough and general description of the process. It might be enough for you, but it also might not. So, and that goes for all these other videos as well. If it's enough for you, great. If it's not, then you know, continue to do some research using your textbook, using YouTube videos, all very helpful, and classwork as well. So let's get into it. The process of translation really occurs in the red here. We have our mRNA strand. All right, so mRNA strand exists. There is always a sequence, a 3-triplet codon sequence that is called a start sequence, and it's usually AUG. So with AUG being what we call a start sequence, the first tRNA, which has its anticodon here. All right, so tRNA molecule has an anticodon, all right, and also has its amino acid attached. All right, so the start sequence, the first anticodon attaches to this codon, the AUG, and the first amino acid to be put on a chain is usually always methionine or met. All right, so remember, we're reading now in the 5 to 3 direction, and the anticodon is again anti-parallel in the 3 to 5 direction, because this piece right here of the tRNA is still RNA. Remember that. All right. The only thing that changes differently is this thing right here. Methionine is an amino acid. So the first thing that's happened, the small subunit binds to the mRNA at the start sequence, AUG. All right, methionine attaches. All right, attaches via covalent bonds, actually, for a moment. The second thing that happens is the large ribosomal subunit now attaches to the mRNA as well, and it acts as a clamp. Okay. Now we have this methionine in here, and in the first site of synthesis of the polypeptide is always going to occur in the place called the P site. We have the E site, the exit site, the P site, the polypeptide site, which the growing polypeptide is on, and when the A site, called the active site, which is going to, the new tRNA is going to be coming in here. All right. So the next step here is we have our first start codon AUG. We have our ribosomal subunits now attached and to the mRNA. AUG with methionine is now attached. All right. The next thing that's going to happen is the next sequence, the next codon, is going to attach a new tRNA with its anticodon for this specific sequence is going to attach with its specific amino acid for the anticodon. All right. At this next moment, all right, the first amino acid is going to be transferred onto this incoming A site amino acid. All right, as you see here. So methionine stays the first polypeptide, but now valine, which is attributed to its specific anticodon, which attaches only with a specific codon, is now a 2-polypeptide long chain. The next thing that's going to happen, all right, is these two, well, actually the ribosome itself will move over towards the 3' direction. So the P site is now going to be the A site, or sorry, what it was in the A site is now going to be in the P site. What it was in the P site is now going to be the E site, I mean the exit site. The exit site is the tRNA that no longer has its amino acid attached. So you can imagine as this ribosome moves over, the P site becomes the E site, all right? The A site becomes the P site, and then now this new codon will become the A site, all right? So as you're seeing right here, the A site amino codon sequence has a specific anticodon tRNA sequence that has its own specific amino acid again. So now we have our exit site, we have our P site, growing polypeptide site, and we have our active site, which is our incoming tRNA. All right. So again, as you see, there is so after all of this is said and done, all right, we do also have a termination or stop codon, all right, called UGA. All right, so our AUG is our start codon, where everything starts, and usually hundreds, sometimes even thousands of amino acids later, or tRNA later, will be a stop codon. So it's not like our polypeptide will only be 3 amino acids. This stop codon, they're actually showing this way down the line here. The stop codon will usually occur hundreds or thousands of amino acid chains later. What we're left with is a polypeptide with a bunch of specific amino acids that will then, on its own, aided by chaperone proteins, fold into correct form depending on what it's needed for. At that moment, once the stop codon and release factor has occurred, all right, the ribosomal subunits will detach, they will release the mRNA, and the polypeptide will get transferred to where it needs to go. All right. So this kind of ends up the process of translation. We can continue on here as well. These molecules right here that look like little crosses, these are the tRNA. The tRNA on one side have their anticodon, which is their triplet code, all right, and on the other side they're actually holding their amino acid. So over here we take a look at that. The anticodon matches with a codon, and each anticodon has a specific amino acid attached. All right. So this anticodon is looking to bind to UAU, all right, which is right here, all right, and this here anticodon is going to bind to UGG, which is right here. So we have our ribosomal subunits. We have our 3 sites here, all right. This is the E site, this is the P site, and this is the A site. Remember, we're reading in the 5 to 3 direction now, so the new anticodon is always going to come towards the 3' end, all right, and the leaving tRNA, all right, it has already put its amino acid on the chain, will exit towards the 5' end of the mRNA. Okay, so this is the process of translation, and it's how we actually translate nucleotide language to protein language, and it occurs in the cytosol. All right, thank you. I look forward to all your questions, and I'll see you later.